Hello everyone and welcome to my channel Science Query. Today I'm going to talk about fat digestion or lipid digestion. Can you explain what is fat in a simple term? Well, fat is a natural oil substance occurring in animal bodies. Chemically, the fats are defined as the esters of fatty acid. The term fat often refers specially to triglycerides, that is the glycerol backbone with 3 chains of fatty acid. Therefore, triglycerides are triple esters of glycerol. Our meal is full of triglycerides. Now let us see the process of digestion of fat and how it happens in our body. Saliva in our mouth moistens the food and this process is known as emastication. The fat present in the food initiates the secretion of enzyme lingual lipase on the tongue. Lingual lipase enzyme is a member of the digestive family. It is also known as triacylglycerol lipase. These lipase enzymes digest lipid. However, this enzyme is still not activated in the mouth. It only cleans the individual fatty acid in the mouth. It gets activated in the stomach in the presence of low pH or in the presence of acidic environment. Now let us understand the step-by-step fat digestion in the human body. First, the saliva of the mouth moistens the food by emastication. Here the enzyme lingual lipase gets associated with the food. The food is now softened and mixed with lingual lipase enzyme and goes in the stomach. In the stomach, the food breaks down further by mixing and churning with enzyme gastric lipase. Therefore, in the stomach, the enzyme lingual lipase and gastric lipase gets activated due to low pH. After mixing with enzymes and churning in the stomach, the food goes into the small intestine, where further fat digestion happens. The digestion of fat in small intestine is done in 2 steps. First is emulsification and the second one is enzymatic digestion. Emulsification is the process where the large lipid globules breaks down into smaller droplet. Here the enzyme pancreatic lipase mixed with the food. The fat droplets and the acidity of the stomach stimulates the small intestine cell to produce 2 important type of hormones known as CCK, cholecystokinin, and secretin. This cholecystokinin stimulates the bile production and secretion. This hormone CCK also stimulates the pancreatic enzyme, that is the pancreatic lipase or pancreatic juice from the pancreas. The other hormone secretin stimulates the bicarbonate ion in the pancreas, which helps in fat digestion by increasing the pH to 6.5. Let us understand the process of emulsification and enzymatic digestion in the small intestine in detail. Large fat globules in the small intestine breaks down into small lipid globules. And during this process, the hormone CCK and secretin stimulates. Hormone CCK stimulates the secretion of pancreatic juice containing enzyme pancreatic lipase. This enzyme pancreatic lipase mixed with the triglyceride, that is the fat droplet, and breaks it down into monoglyceride and fatty acids. Besides the stimulation of pancreatic lipase, the hormone CCK also stimulates the bile production. The bile from the gallbladder does the emulsification process by mixing with small lipid globules, forming micelles. The micelles are the cluster of bile with the fatty acid or monoglycerides. Let us see some of the interesting facts about the bile. The bile is produced and stored in the liver and is secreted in the gallbladder. Bile has the property to interact with the water and fat. After the interaction of bile and fat droplet and the forming of micelles, they started moving at the edge of the microvilli, from where they diffuses into the epithelial cells. Now these fat droplets started breaking into further small drops in the Golgi bodies, where they interacted with more enzyme forming chylomicron. These chylomicrons are the type of lipoprotein. The free bile on the surface of the ileum goes into the liver, from where they are recycled and stored and is secreted again in the gallbladder. This cycling of bile from the intestine to liver to gallbladder and again to intestine is known as enterohepatic circulation. The chylomicron acts like a vehicle. It transfers the lipid from small intestine into the blood vessels. The main function of these chylomicrons is to transport the triglycerides from the intestine to the liver, muscles and adipocytic cells. Well, that is all about a simple description of fat digestion. If you like the video, then please subscribe and share my channel Science Query. Thank you.